How you doing, guys? It's Mr. Shiz. I'm checking in. Today's LEQ is explain why DNA replication is necessary for life. Before we get to that, I want to give you some fun facts about DNA that I think is pretty interesting when you think about something we can't really see unless we extract the DNA, which you're either watching this video and already did the day before, or you're watching this video and you're going to do it the next day. But some fun facts here. So every human being shares about 99% of their DNA with every other human. So that's pretty interesting to think because we all look so different from each other and yet we're 99% of the same DNA. So think about that in the sense that 1% means so much of a difference between every single one of us. So that's a fun fact right there. I got another one coming right at you. So if you put all the DNA molecules in your body end to end, the DNA would reach from the Earth to the Sun and back over 600 times. So that's 100 trillion times 6 feet divided by 92 million miles. So that's a very, very long strand of DNA, to say the least. So that's 600 times back from the Earth and the Sun if we put all those DNA molecules in our body end to end. And that last one here, you have 98% of your DNA in common with the chimpanzee. So that's our closest relative in the sense of a DNA is a chimp. You know, that's what brings us to the sense of evolution, that possibly that we arose from apes. Now, moving on to the first thing we're talking about in today's video. So you got the LDQ there. Make sure you got the top. So first thing we're looking at is the role of DNA. So what does, think of DNA as kind of like a blueprint, right? So it has 3 different roles. And those 3 different roles are to store information, copy information, and transmit information. So we're going to look into a little bit of that today as we go through this replication station here, or not the station, to this video. But first you need to know what the structure of DNA molecule is. And you may have seen this structure before. So looking at this structure, it is a double helix with a sugar phosphate backbone. So that double helix, it kind of just looks like a twisted ladder there. Just seeing that picture. You see those base pairs. We actually will go into that part in a second. But it's a double helix with that sugar phosphate backbone. And remember, we talked about that phosphate backbone when we talked about macromolecules. If you remember that macromolecules, it's a nucleic acid. That Na standing for nucleic acid. Nucleic acid is the only one with that phosphate backbone here. Now, we kind of just got ahead of ourselves, but if you remember that picture you glued in, once again, that macromolecule is a nucleic acid. You see here the phosphate backbone. You have deoxyribose. That's the D in the DNA stands for deoxyribose nucleic acid. And then going to those base pairings that we talked about, it has 4 base pairs. And it has 4 bases. And those are adenine, cytosine, guanine, and thymine. So we're looking at number 4 here on your Cornell notes, but those are the 4 base for DNA. Now they actually have to pair with each other, and you'll see why in a second they have to pair with each other. So here you see the base pairs where it's adenine and thymine bond together, so it's A to T or T to A, and cytosine and guanine bond together, so it's C to G or G to C. So notice on this picture here, thymine's yellow, adenine's pink. Notice that they all go together with each other. The pink and yellows are always bonded with each other, as you see. And then notice the light green and the purple are always bonded together. So the A's can never bond to the C's or the G's, and the C's or G's can never bond to an A or a T. Those 2 have to go with each other for those base pairs. Now, talking about why DNA's replication is necessary for life. So I said before in the beginning of the video, think of DNA like a blueprint. So the cells need to divide before they die, and DNA gives cells instructions to operate properly. So that kind of goes back into the cell cycle. So the cell grows larger, the cell duplicates its DNA, and then that DNA is checked for mistakes. So for those cells to operate properly, to do their function that they are meant to do, that DNA needs to replicate and to make sure that cell actually does what it's supposed to do. Because if it doesn't, then we start having mutations, and those mutations can lead to a lot of harm within human beings and even animals outside of humans in general. So DNA replication is very important for life because without it, your cells are not going to do what they need to be doing. Now, when we talk about how many strands of DNA there is, It's two strands of DNA with one strand being the original and the other is the new strand. So we'll get into that basis of how that actually works. Where like you have one original strand, one new strand, how they all go together. But once again, I just want to make notice here the A's and T's are bonding together. The T bonded to the A, C to G, G to C. So remember, it's very important that you know those base pairs where that A bonds to T or T to A, and then C to G or G to C. Now, we're moving on to the left-hand page, and we're going to talk about the 3 easy steps for DNA replication. And if you understand these 3 easy steps, DNA replication will come very easy to you. So, with that first step there, the enzyme DNA helicase unzips the DNA double helix. So you have this DNA double helix right here. Now, what happens is the enzyme helicase comes in and actually unzips it. So think about it like, it's just cutting it straight in half. So it breaks apart those base pairs. And then once that happens, it goes into step 2. And we have another enzyme that comes in, and that enzyme is DNA polymerase. And that forms a new strand by adding complementary strands. Remember, A to T, or T to A, and C to G, or G to C. So this DNA polymerase comes in, and notice how it's unbroken right here, but as it moves forward, to the right-hand corner, that A will grab a T, that C will grab a G, G will grab a C, and that G will grab a C. So it has that template strand and that new strand because the DNA polymerase comes in and it continually makes another new strand of DNA that goes along with that template strand or that original strand. Now, the third step we're talking about here is kind of like the end result. So 2 DNA molecules are formed that are identical to the old strand. Because remember, they're identical because of those base pairings. So those base pairings stay the same, whether it's A to T, T to A, or C to G, or G to C. Now I have a video here to show you, kind of give you, it's a 3D animation of DNA replication, and we'll watch that to kind of help sum up this video here.
DNA is a molecule made up of 2 strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The 2 strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand, and wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The 2 strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the 2 strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.